Good afternoon. I'm Greg Carty. I'm the Associate Dean for Research in the School of Kinesiology, and it's my pleasure to welcome you today to this special seminar, which is going to be presented in just a couple of minutes by Andy Ludlow, who's an assistant professor in the School of Kinesiology. Andy was awarded the Marie Hartwig Research Grant, which is an internal grant in the School of Kinesiology in 2018. He since has been doing work to support that grant, and he's going to present his findings related to that. Just to give you a heads up, we're going to use the Q&A function, which is down at the bottom of your screen for asking questions, and you're welcome to type those questions in at any time, but we're going to hold the responses from Andy until after he's completed his presentation. So again, we're happy to have Andy present. His topic is exercise in cancer. Is there a telomere connection? So Andy, it's all yours.
Okay, thanks Greg for the introduction. I'm going to go ahead and get started. So my lab here at Michigan was established in 2017, so 4 years ago now, and we've since arriving here, we've started up our research on investigating telomere biology and if there's a connection to aging and cancer. So I'll tell you a couple of different stories in the lab today. First, just some background. I'm sure the word telomere and telomerase is maybe foreign to some of you, but I'll do my best to give you some background about that. I'll talk a little bit about the role that telomeres and telomerase play in aging and cancer. I'll briefly review some of the older studies connecting exercise to telomeres and telomerase. Some of the additional work that we do in the lab focusing on lung cancer and stem cells and how telomerase plays a role in those two contexts. Then we'll go through new data and emerging directions in the lab that we're exploring. So first, our background on telomeres and telomerase and how they play a role in aging and cancer. So first of all, what are telomeres? So in our cells, our DNA is housed on our chromosomes. Our chromosomes are shown over here in this micrograph in blue. And at the very, very tips of our chromosomes, we have these yellowish dots here shown here, or in this cartoon, these red ends of our chromosomes. They're made up of repeat DNA in the structure of, or in the sequence of TTAGGG. So telomeres are extremely important in performing two functions in a cell. As you can see here, if our chromosome ends were exposed as just naked DNA, they would be attacked by endogenous DNA damage sensing systems, and our cells would fail to continue to replicate. So that would be a negative. We wouldn't be able to continue to grow as an organism. However, telomeres and a group of proteins that I'll describe later fix that problem or solve that problem by protecting our chromosome ends and hiding them from our endogenous attack by DNA damage and repair signaling. Further, due to an issue with DNA replication and DNA polymerase being unable to fully replicate our chromosome ends, telomeres shorten every time a cell divides. And over time, our telomeres lose their functions, they're sensed by DNA damage, and this causes the cells to senesce or die. So these are the two major functions of telomeres. One, they safeguard our genome from being attacked by ourselves and destroying our cells. And at the second side of things, they act as kind of a mitotic clock. Every time a cell divides, you lose a little piece of DNA, and it kind of counts how many times that cell is divided. And when it reaches a certain limit, the cell can no longer divide. And so why is that important? So it's been shown that as we get older, our cells need to divide. We need to replace and repair tissues as we age. And what's been observed is that in humans, telomere shortening across multiple tissues, regardless of replicative history, telomeres shorten with increasing chronological age. So in both males and females, you can see there's quite a bit of variability, but the general tendency is that telomeres shorten over time. So there's plenty of research out there on this topic. Recent data indicates that the rate of telomere shortening is what's most important. So even if you have long telomeres to begin with, if you're losing telomere length very rapidly for a variety of reasons, that is a negative outcome. Whereas even if you have short telomeres, but you're doing a good job at maintaining them, that can be looked at as a positive health outcome. Conversely, what happens most of the time is that not all telomeres shorten at the same time. So if we have 92 chromosome ends, so that's 92 chromosomes in a cell, that means 92 telomeres. We can, if only a few are shortening, that's what the cell senses, and that's what can shut your cell down from dividing and thus be a part of the aging process. So the cell senses only the shortest telomeres, and that can induce cellular senescence and aging. Further, short telomeres matter in human health and disease. Over here, there are several genetic disorders with mutations that cause extremely short telomeres, and these individuals have a shortened lifespan. Further, cardiovascular disease, inflammatory diseases, and cancers, they show average telomere lengths in humans, but once your risk can increase if you have extremely long telomeres for certain diseases. And then in other age-related disorders such as aplastic anemia, pulmonary fibrosis, and myeloid dysplastic syndrome, these individuals tend to have the shortest telomeres, thus making telomere length extremely important in human health and disease. So there's been a lot of efforts over the years to try and understand the factors that impact telomere biology with aging and how we might be able to manipulate telomere biology. So there are some exceptions to the telomere shortening rule. Especially important, the male germline cells are immortal. That means that these cells do not experience much telomere shortening, if any at all. And in some instances, you may actually observe telomere lengthening in older males' germline cells. Most cells most tissues are maintained by adult stem cells. And during the development of human fetal development, these cells slow the rate of shortening, and they have delayed replicative senescence. And then in disease, cancer cells have short telomeres, but they have circumvented the telomere shortening mechanism through various means, and these cells are essentially immortal. So what are some of those? What is the telomere shortening mechanism, or what can counteract the telomere shortening observed in some of these cells? So there's an enzyme, telomerase, and I'll talk to you briefly about what telomerase is. So telomerase is a ribonucleoprotein. It consists of a protein component, TERT, and an RNA component, TERC, T-E-R-C, telomerase RNA component. And these two combine as the minimal subunit to form the enzyme telomerase. And the enzyme telomerase is extremely important because to counteract telomere shortening, telomerase de novo adds telomere repeats. So it uses TERC, the RNA template component, to add new nucleotides onto the ends of our chromosomes. And as I just described, only certain cell types have this. So during development, stem cells have abundant telomerase, and they're able to maintain their telomeres even in the face of sustained proliferation. So as our tissues are developing in utero, we have abundant telomerase up until a certain point of fetal development when telomerase is shut off. Then as telomerase shut off, this allows for telomere shortening to occur, which is could be called telomerase insufficiency. So this telomere shortening is not just a mechanism to count cell divisions, it's also an extremely important mechanism to turn off telomerase and suppress the formation of tumors. as cells accumulate short telomeres, they're removed from the dividing population. If a cell were to accumulate with short telomeres and acquire mutations, which would allow it to continue to divide, and it reactivated telomerase, you would end up with a cancerous cell. So in 90% of human cancers, it's been observed that telomerase is turned back on and making the situation very similar to abundant telomerase during stem cells. So once you have telomerase turned on, there's a sufficient telomerase to maintain telomeres. So couple of things to point out here. Telomerase is not tumorigenic by itself. And this has been shown extensively with the use of overexpression of telomerase in normal somatic cells, which once you overexpress telomerase, these cells become functionally immortal, but they do not form tumors. So under most circumstances, in normal cells without oncogenic mutations, telomerase is not enough to form a cancerous cell. So cancer forms once you have enough mutations, along with telomerase reactivation to maintain those short telomeres and allow for immortal growth or continued replication. Okay, so as such, telomeres and telomerase have been touted in the field as hallmarks of the aging process and of cancer. So two cell papers that have come out over the years that have kind of cemented a lot of these ideals and ideas of what aging is at the cellular level and what cancer is at the cellular level as well. And both, so telomeres and telomerase represent kind of a double-edged sword. You want enough telomerase to maintain your telomeres and prevent excessive telomere shortening, and thus you can sustain tissues and maybe resist some aspects of the aging process. But on the flip side, if your cells acquire too many mutations and they continue to proliferate even though they shouldn't be proliferating, and you reactivate telomerase, this can enable replicative immortality, thus giving that yin and yang of how much telomerase do you want and how tightly regulated do you want telomerase. So you want a telomerase to be on when you want it on to maintain telomeres in normal cells or normal stem cells so that you can repair your tissues sufficiently. Whereas in cancer cells, you really don't want a lot of telomerase because you want to get rid of your cancer cells. You want to essentially make them mortal again so that they do not continue to divide and form tumors. So this is where the crux of my research comes in to date. So what's known about connecting exercise to telomeres and telomerase? So a brief review of some older studies. So I have a question up here. It's a very broad question. People have been trying to address it for a long time. And does exercise simultaneously slow aging and reduce cancer risk by slowing telomere shortening? So what do we know about exercise? This is a kinesiology audience, so I don't really have to go into too much detail here. I think exercise enhances immune cell function and potentially enhances immune surveillance of different rogue cells, either cancerous cells or senescent cells, lower psychological stress for sure, better neuroendocrine control, metabolic control, a lower need for insulin. There's better regulation of sex hormones if you're a physically active individual as you age. There's increased antioxidant defenses and reduced inflammation. And all of these factors combined can lead to successful aging. So this gentleman over here recorded the fastest 100-meter dash a few years ago now, but he was in his late 90s when he did this, and he looks quite robust for his age. So what is known about telomeres and physical activity, and what is the relationship? So several papers in 2008 addressed this topic in a variety of fashions. So a group consisting of researchers in the US and in the UK took a large cohort that consisted of non-related individuals and a twin cohort. And what they observed was this linear relationship between sedentary behavior all the way up to being pretty physically active. And you can see large numbers of individuals in each of these groups. So this correlation was quite strong that individuals in the heavy and moderate group had longer telomeres than the inactive group. So this paper came out in 2008, right at the same time that I was completing my master's thesis. And what I had access to was a cohort of about 70 individuals in the age range of 50 to 70 years, some of which about 20 of these individuals were masters athletes compared to individuals that performed various amounts of physical activity. So we had quartiles of individuals. We had access to peripheral blood mononuclear cell DNA, and we were able to measure telomere length across these groups of individuals. So we didn't directly measure their physical activity, but we were able to use a questionnaire to get an idea. And we parsed these individuals out into quartiles and observed that moderately active individuals had longer telomeres than these sedentary folks and folks that were extremely active for a long period of time. So I don't want to make the statement that physical activity is bad for your telomeres, because I don't believe that. I think that there's maybe these individuals, the cumulative stress of being highly physically active throughout their life may have blunted their telomere or enhanced their telomere shortening rate in these immune cells. But at the same time, there may be mechanisms that these individuals had that counteracted that in other functional fashions. So recent studies have looked at exercise training. So aerobic and interval training increased telomerase and telomere length in middle-aged adults. So I think a lot of the literature that explored telomere biology and exercise in young individuals has missed an opportunity, because I think young individuals have robust stress responses and they're able to maintain their telomeres. Their telomeres aren't shortening very rapidly. But as we age, and I think in midlife, especially that 45 to 55 years of age, lifestyle starts to really catch up to these individuals. And that's when physical activity can really show a benefit, especially when it comes to telomerase activity and telomere length. So what this recent intervention study showed was that aerobic endurance exercise, a single bout of aerobic endurance exercise resulted in an increased amount of telomerase enzyme activity, and while resistance exercise training did not. So these individuals measured telomerase activity in their peripheral blood mononuclear cells at 3 different time points following those exercise bouts. And the general gist of it is that aerobic exercise, not resistance, resulted in enhanced telomerase activity. And then in a training study, these individuals were trained over several weeks and months, and they looked at telomere length changes pre-post the intervention. You can see no real change in the control group pre-post. However, in the aerobic and interval groups, you saw telomere lengthening or enhanced telomere length in the aerobic and interval training group versus resistance training. So this is also confirmed by another study. So that was in blood cells, in a study that looked at telomere length in skeletal muscles with aerobic exercise training. So they found that 12 weeks of exercise training, aerobic exercise training, was enough to elongate telomeres regardless of placebo or metformin group. The pre-post was significant. And these again are that 63-year-old individuals, again pointing to lifestyle factors becoming very important as you reach midlife, especially for telomere biology. So this is kind of the background for why exercise and telomere biology may be extremely important for slowing the aging process, both in blood cells and in skeletal muscles. So the summary to date in exercise, I think exercise is related to changes in telomere length. That the safest conclusion is that moderate exercise maintains telomeres. Telomerase may be activated as an adaptation to slow down the rate of telomere loss. But I think there's still a lot of area to be a lot of questions to be answered here. So what are the mechanisms of these adaptations? And during my postdoc and since arriving here, we've improved upon the measurement technologies and methodologies for telomerase and telomere length. So I think applying some of those techniques to exercise trained either humans or animals may lead to new conclusions. So before we dive into that, let's ask the question of how is telomerase regulated in humans? So we know that the function of telomerase is to maintain telomeres, but we're unsure about the exact mechanisms that regulate telomerase. So something that's been studied for a long period of time is the transcriptional control of specifically the telomerase reverse transcriptase component, or TERT. So just a brief review, telomerase consists of two core components, the protein catalytic component, TERT, and the RNA component, TERT. So most of our cells have sufficient levels throughout our life of TERT. However, when cells, after through development and we become somatic cells, most differentiated postmitotic tissues turn TERT down via transcriptional control, and this transcriptional control silences telomerase. So most of our adult tissues are telomerase negative, and the rate limiting component is mainly TERT RNA expression. So while the transcriptional control has been studied in how it's reactivated in cancer, very little is known about other aspects of controlling telomerase levels, and one of those is the RNA metabolism. So RNA metabolism is a variety of different processes, alternative RNA splicing being one of them, along with 3' UTR processing. And this area of RNA metabolism and how TERT is controlled by these processes is largely uncharted territory. So what we've done is found some novel finding, and potentially a paradigm shifting finding, was that hTERT is transcribed in most cells, but this transcription was processed by alternative splicing to variants that didn't form telomerase. And this allows us to understand alternative splicing and maybe develop targeted therapies to maintain telomeres or shorten telomeres in cancer. So what is alternative splicing? Alternative splicing is a gene regulatory process. It generates proteome diversity and regulates functional outcomes of gene expression. So how does alternative splicing work? So our genes are on our chromosomes, and they're organized in exons, shown here in the blue, green, and red boxes, separated or interspersed by intronic sequences. So you have an exon, intron, exon, intron, exon type of setup. And there are two different features, one in cis, and cis refers to the sequence elements, and trans refers to things that bind to those cis elements. So what you get is splicing being regulated by proteins that are both, there's a core set of splicing factors called the spliceosome, and there's 300 of those. And then there's an additional about 1,200 tissue-specific splicing factors that can act as enhancers or repressors of that spliceosome being assembled to come in and cut out these intervening sequences to make fully functional mRNAs. And these two isoforms could be the result of alternative splicing. So if this is the full length, all exons included, this would be considered your spliced isoform. And then when you exclude this green exon and you just have the blue and red, you get an alternatively spliced isoform. So there's lots and lots of ways that this is regulated, such as sequences near the exon-intron boundary, sequences more internal to the introns, and also the way the RNA folds after it's transcribed. So I'll go through a little bit of what we know about TERT and what we've discovered in my lab here about alternative splicing regulation of hTERT. So the general idea is that hTERT is a 16 exon gene. We study a critical splicing event in here, where if you include these two exons in blue, that they generate a full-length telomerase molecule, and you could imagine that this could be useful for rejuvenation therapy or adding telomere repeats to aged cells so that these cells can function better. And conversely, when telomerase, this goes on to form telomerase. Conversely, when these two exons are skipped, you end up with an alternatively spliced isoform that does not form telomerase. And you could imagine wanting to block full length and promote this splicing event as a cancer therapeutic, and that could potentially shorten telomeres and limit the growth of your cancers. So some of the key questions that we ask in my lab are, what are the cis or sequence elements that dictate hTERT splicing? What are the proteins that regulate hTERT splicing choice? And a new avenue that we're headed towards is, what are the functions of these hTERT splicing variants in various physiological contexts? So a recent paper from my lab, from a postdoc in my lab, Aaron Slusher, what we ended up doing was we utilized a large cohort of RNA sequencing data to investigate what was the most common hTERT splicing event across 31 different human cancers. And these are the cancers are shown down here. So this is called the Cancer Genome Atlas. And these are patient tumor samples that have been sequenced via RNA sequencing. And then you can determine what splice variants are expressed across various genes. And so since we're interested in TERT, we studied the hTERT isoforms that were expressed in each of these different cancer types. And what we did was we compiled the profile of each of these different isoforms. So TERT forms a multitude of isoforms, full length being the one that contains all 16 exons, and it forms your active telomerase. The majority of the other proteins are either dominant negative isoforms, meaning they counteract telomerase's action at the telomere, versus some of them are just mostly degraded by a process called nonsense-mediated decay. And all of this combined means that we only want a certain amount of full-length telomerase in our cells, and alternative splicing is one way to ensure that. And even in cancer cells, that's important because the regulation is still intact. So as you can see, this orange bar represents any splicing event that contains the skipping of exons 7 and 8. So boxed here in this red box are exons 7 and 8. And you can see that across all 31 cancers, that this event is one of the most common splicing events. So these three isoforms end up making up the majority of the transcripts that are made, indicating that the functional amount of telomerase, this blue bar full length, is relatively a minority percentage. So my lab studies lung adenocarcinoma, and you can see it sits around 24-25% full length, with the rest of these transcripts being alternatively spliced to either dominant negative or non-functional forms, the most of which contain this minus beta splicing event. So we thus focus on the inclusion or exclusion of exons 7 and 8, and through a series of molecular biology papers that I won't go into the details, we have discovered a cis element that is found in the deep intronic region between exons 8 and 9. And this region we call direct repeat 8, and it's conserved in these old world long-lived mammals. But you can see down here in mouse and rats, they don't contain this intronic element. And so the function of this has been elucidated by a publication that we had in my lab come out in 2019. Former postdoc in the lab, Mohammed Saeed, and myself completed the majority of this work. And what you can see is that what this data shows from long read full length RNA sequencing data, where what we were able to do was sequence from exon 2 to exon 16 and very carefully quantify the number of transcripts. So if you see a blue, that exon, or a green, that exon is included. The white means the exon was skipped. And you can see in humans, so these are HeLa telomerase positive cancer cells, the majority of the transcripts have these white dots in them, meaning they're missing critical exons and they can't go on to form telomerase activity. So the second most abundant isoform was this full length. And down here in a NIH3T3 mouse fibroblast line, we compared this to what the isoforms were from a mouse. And you can see the majority of the transcripts are this full length containing all exons of isoform. So this is just recapitulated over here on this RT-PCR gel. The human cells, HeLa, right here and here, you can see clearly this minus beta splicing event shown by this full length band is here and a minus beta band shown here. Whereas in the mouse cells, the darkest band here on this gel indicates full length mouse TERT. So we think that what this indicates is that humans use this intronic element to regulate, very carefully regulate how much telomerase their cells can produce. And mice, since they're short-lived, have a different use an early reproductive life strategy. They don't care about cancer prevention as much, whereas humans, which are longer, much larger organisms, they have to prevent cancerous growth. So our somatic cells shut off TERT so that we can grow and divide without the risk of transformation in cancer. So continuing on, building on this idea that telomerase is regulated by this intronic element, we have built through a series of 3 papers. Aaron Slusher, again, postdoc in my lab, has discovered now the 3rd RNA binding protein that we think interacts with this DR8 element. So we've previously described 2 genes, NOVA1 and PTBP1, which bind this DR8 element and promote the inclusion of these 2 exons and promote telomerase enzyme activity. Recently, we found that this other factor, SF3B4, also binds at this region in both NOVA1 positive and NOVA1 negative cell contexts to promote telomerase enzyme activity. So just to further elucidate the importance of this, we utilized a CRISPR-Cas9 strategy to either mutate or delete the DR8 region. So we used, in this strategy that I'm showing here, we mutated it. So for those who don't know, CRISPR is essentially a molecular scissors. You can go into cells with CRISPR and cut the DNA, and then you can add in pieces of DNA of your interest that you can then use that to study the function of that piece of DNA. So we used CRISPR to insert a wild type, but that's our control, wild type DR8 sequence in the endogenous hTERT locus of these H1299 cells, versus we also mutated DR8 with these, we mutated the NOVA1 motifs, but just DR8 mutant is all you need to know. So moving forward, to kind of use a sledgehammer approach, we also cut out DR8. So we used 2 different CRISPR guides to completely remove DR8 rather than just mutate it. And now this generated an altered hTERT gene allele. And we could then ask the questions of what in cells, in cancerous cells, what is the role of DR8 in maintaining telomeres, producing TERT full-length transcripts versus spliced, alternatively spliced transcripts, et cetera. So what we observed was quite striking, was that so we had multiple different types of controls. So this is your control, potential full-length versus alternatively spliced TERT, and you can see the ratio here. And then when you mutate or delete, you end up with only alternatively spliced variants of hTERT at the mRNA level. And further, what that does to the telomerase enzyme activity, we show that the mutant cells have much less telomerase activity compared to your two controls over here, and the deleted lines have even less. And this is reflected in a functional outcome of the telomere length of the cells. So very briefly, I'll go through how to measure telomere length. So the DNA is extracted from cells, it's digested with restriction enzymes and then run on a gel and transferred to a membrane whereby we can probe that membrane for telomere repeats. And so the early passage cells and late passage cells of the parental, you can see that this smear is not shortening. It's not going any shorter. Same for these sorted controls. The smear is maintained. It's not shortening. However, if you look at the mutant and the deleted lines at early growth versus later been passaged and divided many, many times, you can see that telomeres are shortening. And in the deleted lines, you can see these telomeres. Sorry about that. These telomeres are extremely short. So what was most striking was even just by deleting this intronic element, we observed that several of these clones did not survive in tissue culture. So they died eventually, indicating the importance of this intronic element in producing sufficient telomerase to maintain telomeres and immortality of those cancer cells. So to move this work forward, Aaron, when he joined the lab, set up a series of xenografts. And so we took those H1299 lung cancer cells and injected them into the hind flanks of immunocompromised mice. And so these are representative images. We had 6 tumors, 6 injections per tumor type per group. So six tumors were potential from the population control from each of these different cell lines. And what this result was extremely striking to us was that the mutants, all but one tumor, so we had 1 out of 18 injections, we only found 1 tumor. And in the 2 deletion lines that we tested, we found 0 tumors, indicating that just by disabling telomerase with this intronic element, we were able to prevent cancerous growth in this model system. So this was exciting to us, and so we had some questions and concerns because, as I've told you, there's telomerase in our stem cells. We might need telomerase for more tissue maintenance than in cancer cells. However, telomerase is an attractive cancer therapeutic target. It has met many clinical barriers. So we wanted to see if the work that we were performing at the basic science level was also relevant in stem cells. So we asked the question, how is telomerase turned off? And are there similar or different factors involved in stem cells versus cancer? So we're in the beginning stages of this work. And I'm not going to go through all of the data here. I'm just going to give you our working model of what we think is going on. And so my PhD student, JJ Kim, and former postdoc who started the project were working on this with me. And so our model that we have some pretty good data for indicates that a protein factor, which we also observed in cancer, PTBP1, binds this DR8 element and promotes the formation of full-length TERT and telomerase enzyme activity. And we're studying this in human-induced pluripotent stem cells. So what we can do in the lab is we can take these induced pluripotent stem cells and convert them into various different cell lineages. So through the differentiation process, just like tissue specification during development of a human, you would get telomerase shut off, turning down. So that mechanism is twofold. One is a transcriptional response, and the second is you have to very fine-tune the telomerase activity that's left in these differentiated cells by alternative splicing. So we used a model of turning these induced pluripotent stem cells into neural progenitor cells. And another one of our factors, NOVA1, is a well-known neuronal specific splicing factor. And so this NOVA1, we've observed interacts with a homolog, or a different protein very similar to PTBP1 called PTBP2. And we think that this protein combination at DR8 actually displaces PTBP1 and results in telomerase being turned off. So we're in the midst of testing these hypotheses in the lab, and I just wanted to share a little bit of that data with you now. So, however, what we've done in terms of the Hartwig Award and exercise and how telomerase and shorten may respond to exercise, I'll dive into now. So I've just spent about 20 minutes telling you how different humans and mice are for TERT regulation and telomerase regulation. So how can we perform studies in difficult to get tissues and try to understand gene regulation of TERT and telomerase in vivo following exercise? So thankfully, in 2005, some researchers at the NIA developed a very exciting model, at least for me. They took a mouse and essentially humanized this mouse model, and I'll explain what that means. So mice, as I stated before, do not regulate TERT and telomerase the same way. They keep TERT on in the majority of their tissues, whereas in humans, with the exception of testes and lymph and a few other cells that have a few telomerase or TERT transcripts, are mainly TERT negative and have no telomerase enzyme activity. So the question that the researchers asked initially with this mouse was, is it something about the mouse, or is it something about the human gene sequences that are causing this difference in expression pattern between the two organisms? So what they did was they inserted 54 kilobases of the hTERT gene containing all 16 exons and 15 introns. And what they observed was that once that hTERT gene was in the mouse cells, it followed the same expression pattern. And what this indicates is that the sequence elements are critical, and that the transacting factors, the factors that bind those sequence elements, are highly conserved between the two animals. So even the mouse proteins were recognizing the human gene and treating it like the human gene. So it recapitulates the testes have hTERT and the thymus have hTERT, but the majority of these other tissues do not. So we purchased this mouse and started breeding it in my lab a few years ago. But first, I wanted to. We purchased this mouse a few years ago, and we wanted to ask the question of, can we repeat the data that was published in 2005? This mouse is new to our lab, and what we were able to observe was that the hTERT back mouse recapitulates human alternative splicing patterns across multiple tissues. So we measured both the mouse TERT from the exons 5 to 9, and then we also measured the hTERT looking at the same region. And this gel is important because it shows that our primers are specific to each species telomerase. So this is a no template control, blank, and you can so the reaction controlling that the reaction is clean. This is a human cancer line. You can see that only the human primers detect signal of potential full length in the minus beta transcript. Up here, you can see the mouse is negative for the human primers, and converse, or thankfully on the other side, the mouse primers detect the NIH3T3 mouse TERT and do not detect the human gene. So this gives us the ability to measure human versus mouse TERT in the same tissue. So we then went across and looked at a variety of tissues to just qualitatively detect TERT transcripts in these tissues, and we saw some expression, but we wanted to more carefully quantify this. So we use droplet digital RT-PCR, and this is again performed by a postdoc in my lab, Aaron Schlusher, and he was able to quantify both mouse TERT across these different tissues. So showing that our droplet digital PCR primers are also very specific. The HeLa cells do not pick up any mouse transcripts. The NIH3T3 cells do. And then you can see TERT is expressed at various levels across different tissues. And then over here, we used primers to detect TERT both from the 7-8 exons and exon 6-9, so measuring 2 different splice variants. And again, you can see specificity of our primers where we can detect only the human RNA, but not the mouse. And then you can see the quantification showing the telomerase active tissue has the most TERT transcripts, whereas these telomerase negative tissues show much lower, much more repressed levels. So having a model in hand, we wanted to ask the question of what does an acute bout of exercise across several tissues, how does that impact TERT expression and splicing pattern? So we set this up. So I think of exercise and maybe in a little bit of a different context, but I look at it as a physiological stress. So does physiological stress increase hTERT expression? And so to do this, we use a rodent treadmill shown here, and we did an acute exercise model. We bred out the animals so that we had about 5 animals per group, 2 to 3 males. There was the acclimation period, a speed test so that we could determine how fast each animal should run. And then they performed a single bout of exercise on day 14. So on day 14, then the animals were sacrificed at various time points post that bout. So immediately post, 1 hour post, 24, 48, and 72 hours post. And then we extracted tissues. I'm going to show you data from the gastroc, the left ventricle, and the brain briefly here. So what we were able to see was that exercise-induced splice switching towards full-length hTERT. So again, this is a gel showing the human gene being expressed from exons 5 to 9, so encompassing the potential full-length molecules that could go on to encode for active telomerase, and the minus beta variant that is mostly of unknown function to date, especially in normal tissues. So again, our control here showing that our reaction is clean, and then you can see splice switching and increased in transcription across these different conditions, especially in the gastroc, in the left ventricle at that immediately post time point, with things returning back to baseline levels out here through the time course. And really, there's not much of a change in the brain qualitatively. So again, that from the gel-based assay, we wanted to quantify this much more carefully using droplet digital PCR assays. So I'm showing you data from hTERT exon 7-8. And what you can see was we were able to detect significant differences in skeletal muscle and cardiac muscle here immediately post the exercise test. And so these data indicate that the mice are able to somehow transactivate and splice the hTERT mRNA, pre-mRNA, as it's in the mouse muscle and cardiac tissue. So when we quantified the response of the brain, we saw really no changes as expected. So this led us to a brief hypothesis here, and I'll wrap up on this slide and skip to my future directions. So we have an integrated cellular stress response, and I am hypothesizing that hTERT could be a reporter. So we used young animals to do this, and I think that exercise, on a custom exercise, a pretty high intensity exercise, is an adaptive stress to these animals. So we're going to explore the activation of certain pathways, NF-kappa beta, IL-6, TNF-alpha, which from the cancer literature are known to activate these transcription factors such as ER-alpha, STAT3, SP1. We hypothesize that some of these may be related to increased expression of hTERT, and that then this molecule has to be spliced, and we're going to explore which splicing factors may shift the splicing of hTERT towards either the full length or the minus beta. And then It's well established in the field that full length is going to help maintain telomeres and protect telomeres. But what these other molecules could be doing in terms of mitochondrial protection or DNA repair are really in their infancy. And there's some hints that TERT is in the mitochondria. There's some hints that TERT may interact with DNA repair, but there's really no solid evidence that these things can reduce aging. However, I do believe there's a maladaptive stress response that could be investigated in this mouse model as well, either with extreme exercise stress, aging, DNA damage, or maybe even certain disease states such as diabetes that induce chronic levels of inflammation, maybe mediated by TGF-beta. This can activate central integrated cellular response proteins such as ATF4. These would repress TERT, and then that would lead to mitochondrial dysfunction, DNA damage, etc. So I'm going to skip to my couple of slides ahead and show you why I think this is important. So in recent studies, or for a long time in the literature, everybody thought that telomeres and telomerase only mattered for replicating cells. But recently, 2 reports have come out, one in skeletal muscle and one in cardiac muscle, indicating that there are these TAFs, or telomere-associated foci, in the myocardium proper, so, and in the skeletal muscle, not in the satellite cell nuclei, in the muscle fiber nuclei. And this is indicative of persistent DNA damage, which is a feature of cellular senescence. So I think this interesting finding opens up a new avenue for my lab to potentially test the idea that exercise may be able to prevent some of this telomere damage and senescence-associated phenotype in these post-mitotic tissues via the activation, transient activation of TERT. So what are, in summary, I think that the data are pretty consistent that exercise maintains telomeres across a variety of human and mouse tissues, especially in immune and contractile tissues. The mechanism is likely a transient increase in telomerase. There's probably a multitude of gene regulatory mechanisms that are resulting in this, from transcription and splicing. I didn't get to show you this data, but shorten expression also increases with training and maybe enhances recruitment. And so moving forward into the future directions, there's some, I think, outstanding questions. So what is the function of these full length and alternative splice variants of TERT in somatic cells under stress? What is the role of hTERT and HTR in protecting post-mitotic tissues from senescence and stress-induced senescence? Can we utilize TERT identifier? So can we deliver TERT to these tissues to aid in the regeneration process and rescue these telomere-associated foci? Along with several other exciting avenues that we're considering exploring in the lab in the future. So with that, I've got to acknowledge my funding, especially the kinesiology with the Marie Hartwig pilot grant, which funded the acquisition of the hTERT back mouse and the work to date on our rodent models and exercise. Mohammed, Aaron, and JJ, and Mark, I couldn't do the work without them in the lab. So with that, I'll finish up and take any questions.
Okay, I have to see. All right, great. Thank you, Annie. Appreciate that very interesting talk. So again, we have several questions in the Q&A already, but those of you who have been thinking about it, go ahead and enter your questions and we'll get started, and don't hesitate to put your questions in as you're going. So the first one is from Edwin Miranda. So it's sort of a series of questions. I'll sort of go through them. Does telomere length in different cell types and tissues have different physiological consequences? For example, does increased telomere length in PBMCs tell you something only specifically about immune function, or does it tell you something about the organism's health as a whole? And then just the last part is, could telomere length in, say, skeletal muscle have different implications than telomere length in PBMCs?
So, I mean, Edwin, that's a good question. It's one that I've tried to, I've gotten a lot, and I think it's an open question in the field. We know that, for instance, if you take a disease model and that has short telomeres, and you add telomerase to that disease model, it reduces the severity of the disease. So, for instance, if a heart undergoes a myocardial infarction, that's associated with ischemic damage, and the telomeres get shorter. If you add adenovirus TERT to those animals after the myocardial infarction, it elongates the telomeres, and the scar tissue formation is less. So these animals have a healthier heart afterwards. So that's one functional outcome of elongating telomeres. In terms of PBMC telomere length, I think it's touted as an overall indicator of the stress that an organism is under. It's really a sample of convenience from humans, for sure. And that's where the majority of the studies have been done. But I would suspect that you could make some inferences, but they may not be perfectly accurate in all contexts, from PBMC telomere length to, say, skeletal muscle telomere length. So if an individual is living a sedentary, unhealthy lifestyle, all of their telomeres are probably shorter in all of their tissues. But if you have an individual that's maybe there's some changes in maybe an injury to a skeletal muscle, you may get some differences between PBMC and skeletal muscle telomere length. But I think so you might have to get into the context there. I hope that answers your question.
Great. So then the next question is from Jake Haas. He says, is telomerase regulated by any antioxidant response element targets, being that exercise is a potent stimulus of these genes, these antioxidant response element genes? Similarly, are there any critical thiols within telomerase that are reactive oxygen species sensitive?
So I'll start with the easy part, because the thiols in telomerase, the protein, I think, so the problem with even trying to figure that out from an endogenous TERT standpoint is we don't have antibodies that are good enough to pull TERT down to actually measure it. TERT's very low abundant. From an overexpress standpoint, we might be able to do something like that, where we overexpress TERT and then look at how its protein is modified under different reactive oxygen species conditions. And in terms of the TERT gene being regulated by AREs, I think that it would depend on how much ROS there is. And to date, if I go back to my suspected proposed model here, Jake, on this slide, I think what you could imagine is if the ROS is just right and it's a young, robust, resilient animal, you might get some of these transcription factors and signaling molecules activating these downstream. As for an ARE element in the promoter of TERT, to my knowledge, there isn't one. But clearly, these proteins can all be activated. These signaling proteins are all activated by ROS. So there could be some downstream consequences.
Okay, so the next question is from Mike Bessia. He says, great talk, Andy. Does the type of exercise matter? How about intensity?
So I think the Werner et al. paper that came out in 2019 solidified that aerobic exercise training is probably the most beneficial. And there's also been some epidemiological evidence out there from like the N. Haynes data that asked, if people spent more time walking or running or cycling, which one of those resulted in telomere length maintenance? And it's pretty consistent that aerobic exercise, and specifically running, is better for telomere length maintenance for some reason. I don't know why, but and then the other part of your question, Mike, was, does intensity matter? And I don't have a good answer to you, but that's what this model system that or this model that I'm trying to put together would suggest that there's probably a threshold where you get activation of TERT, and then a threshold where you repress TERT to protect the cells from transforming. If there's so much damage from the stress, you don't want that cell to continue to replicate. So I think that's where you get the adaptive versus maladaptive stress response. And I think that's a really interesting, exciting question.
Okay, so the next question is from Mike Schley. He says, thanks for presenting a complex topic to in an understandable form. Do you have any plans, or can you make a knockout of the humanized mouse model, such as PTB1, NOVA, or DR8 knockout, to show the exercise response altering activities or transcription of these genes?
So Mike, that's a great question. I just talked with Aaron about, we could certainly breed this animal onto any number of, say, in muscle-specific inducible knockouts. A lot of these splicing factors are embryonic lethal, so you'd have to build the system to knock the gene out specifically in muscle and then go from there, or somehow induce it. But yeah, I mean, I think that would be a very logical next step, would be to knock out these genes, put the animal back under physiological stress, and see if you lose the TERT response.
Okay, so I have sort of another question from Edwin Miranda. Thanks for answering my first question. That makes sense. Related to Dr. Haas's question, if TERT is responsive to stress, does that mean that telomeres are negatively influenced by stress? For example, can increased or persistent oxidative stress threaten telomere length independent of that which occurs as a result of cell division?
Absolutely, Edwin. So part of my dissertation, I took muscle fibers from mice and I treated them with reactive oxygen species, and I showed telomere shortening in a very rapid, short period of time. So telomeres are, they have a triplet G, and guanosines are more sensitive to oxidative damage compared to your other nucleotides. That's one point. The second point is telomeres are also refractory to repair, meaning that once your telomeres are damaged, they persist, and that absolutely can result in telomere shortening independent of cellular replication. And these types of questions, Edwin, are just coming on board in the field of aging, because everybody is focused on replicating cells and telomeres and kind of ignored the postmitotic tissues.
Okay, so this next question is from Jesse Pollins-Boyd. Are telomerase abnormalities more commonly caused by mutations of cis or trans acting elements? Is the research of one of these elements more accessible than the other? And then he says, then the next part is, and is the expression of the trans acting proteins further regulated by other cis trans elements?
Okay. So, okay, Jesse, I'm going to answer the first question. I'm going to come back to this slide. So this is the telomerase RMP. So the mutations that the telomeropathies or telomerase genetic diseases, some of them arise from mutations in TERT, the protein component. Some of them arise from mutations in the RNA component TERT. And then these accessory proteins, which I don't have named here because we don't study them in my lab, but some of them are such as discarin, which is an RNA binding protein that tethers TERT to TERT. There are genes that cause dyskeratosis congenita. Those are mutations in the discarin gene, and they result in telomerase abnormalities. So it's the most of the mutations detected to date that cause telomerase issues, telomerase diseases, telomerase associated diseases, are in the telomerase protein itself. So in terms of cis or trans acting elements, I think that's in terms of what I showed to date, there's really not much known. The binding proteins such as NOVA1, PTBP1, SF3B4, they're in every cell. They perform vital functions in terms of handling RNA. So they're not really mutated to my knowledge, and not specifically for telomere biology. I hope that should answer his question.
Okay. And so, and I just want to say, so I got some great questions. It was a really interesting talk. Is again, this is really a pilot grant that Andy was talking about, the mouse model, humanized model especially, that was supported by that. I know we did other work that was supported by other sources, but I guess I was really excited to see that he's gotten some nice data out of that small pilot grant. He also has got some training for a postdoc, a good thing for that as well. And it looks like he's got some exciting questions to follow up. So that's exactly what we want to see from the Hartwig. So I want to congratulate Andy on all he's accomplished so far on this project. I want to thank everybody who's joined us and asked questions. And I'm sure if anybody wanted to email Andy, he would answer any question you might have. And thank you, and have a wonderful rest of your day going into the weekend. So take care. Goodbye.